Namaskar. In this lecture, we are going to understand about the DNA isolation with respect to their procedure and their quantification. Let's start our lecture. So first, we are going to understand about the introduction of DNA isolation. Now here, the DNA isolation process is carried out by mainly the combination of physical and chemical methods. The first time DNA was isolated by the Friedrich Mischer in 1869, and the scientists used DNA in number of applications such as introduction of DNA into cells and animals or plants or for diagnostic purposes. In medicine, the latter application is the most common, and the DNA isolation or identification is mostly used in the forensic sciences for the identification of individuals' DNA. For example, rapists, preventives, accidents, or was. Presence of proteins, lipids, and polysaccharides and some other organic or inorganic compounds in the DNA preparation can interfere with DNA analysis methods. There are different common sources for DNA isolation, which includes blood, hair, sperm, bones, nails, tissues, blood stains, saliva, buccal swabs, epithelial swabs, cells, urine, paper cards used for the sample collection. Bacteria, animal tissues, or plants also used. Now, procedure. Now, see, the isolation of DNA basically consists mainly four major steps. First is the preparation of cell extract. Second, purification of DNA from cell extract. Third one, concentration of DNA samples. And fourth one, measurement of purity of DNA concentration. Here in the first step, where we have to prepare the cell extract, and here we have to purify the DNA from the cell extract, and that purified DNA we have to concentrate with the help of different chemicals, and then at last we have to measure the purity of DNA concentration. Let's see the first procedure, that is the preparation of cell extract. Now here, to extract DNA from cells or tissues of interest, the cells first have to be separated and the cell membranes have to be disrupted. Now the extraction buffer helps in carrying out the procedure. Mainly chemicals such as EDTA, which stands for ethylene diamine tetraacetate, which removes Mg, that is magnesium ions, that are essential for preserving overall structure of the cell membrane, and SDS, which stands for sodium didosyl sulfate, which aids in the disrupting the cell membranes by removing the lipids of the cell membranes, are included in the extraction buffer. So mainly extraction buffer is made up of EDTA and SDS, which are helpful in the disruption of cell membrane. Having lysed the cells, the final step in the preparation of cell extract is removal of insoluble cell debris. Cell debris and partially digested organelles, etc., can be pelleted by centrifugation, leaving the extract as reasonably clear supernatant. Next is the purification of DNA from cell extract. Now here, in addition to DNA, the cell extract will contain significant quantities of detergents, proteins, salts, reagents used during cell lysis step and RNA. A variety of procedures can be used to remove these contaminants, leaving the DNA in a pure form. The most commonly used procedures are ethanol precipitation, phenol chloroform extraction, and the mini column purification. In the ethanol precipitation is mainly used by ice cold ethanol or isopropanol. Here, DNA is insoluble in these alcohols. It will be aggregate together, giving a pellet upon centrifugation. Precipitation of DNA is improved by increasing the ionic strength, usually by adding sodium acetate. Second method is the phenol chloroform extraction. Now in this method, phenol denatures proteins in the sample. After centrifugation of the samples, denatured proteins stay in the organic phase, while aqueous phase containing DNA. The third one, mini column purification. Now in this, relies on the fact that nucleic acid may bind or absorption to the solid phase, that is silica or other, depending on the pH and the salt concentration of the buffer. So these are the three steps used. Next, that is the concentration of DNA samples. Now here, the most frequently used method of concentration is ethanol precipitation. In the presence of salt at temperature minus 20 degrees Celsius or less than that, absolute ethanol will efficiently precipitate polymeric nucleic acids. With a concentrated solution of DNA, one can use a glass rod to pull out the adhering DNA strands, while for dilute solutions precipitated, DNA can be collected by centrifugation and redissolving in an appropriate volume of water. So in this way, we can concentrate of our DNA. Next one, that is the measurement of purity of DNA concentration. Now here, DNA concentrations can be accurately measured by UV absorbance spectrometry. The amount of UV radiation absorbed by a solution of DNA is directly proportional to the amount of DNA sample. Usually, absorbance is measured at 260 nanometer at this wavelength, and absorbance 1 corresponds to 50 microgram of double-stranded DNA per ml. With a pure sample of DNA, the ratio of the absorbance at 260 nanometer and 280 nanometer is A260 A divided by A280 is equals to 1 is to 8. Ratios of less than 1 is to 8 indicate that preparation is contaminated either with protein or with the phenol. So this is all about the DNA extraction by different methods. Thanks a lot.